Hello, my name is Omar Nadeem from the Myeloma Program at the Dana-Farber Cancer Institute, and our topic today will be bispecific antibodies in multiple myeloma. These are my disclosures. So it is safe to say that the available therapeutic options for patients with multiple myeloma continue to accelerate at a very rapid pace. We've had our traditional agents available for a while now, such as immunomodulatory drugs, proteasome inhibitors. In 2015, we got our first approval for monoclonal antibody agents such as elotuzumab and daratumumab, and these combinations are now widely used in patients with newly diagnosed multiple myeloma and relapse and refractory multiple myeloma. Over the last few years, we've had even more approvals in the field of immunotherapy, and this has included two CAR T cell products, and more recently, the first bispecific antibody ever approved for multiple myeloma called teclistamab. And this is adding to the numerous agents available for patients that have had prior therapy with some of the agents that I've just described. So today we will go over some of these immunotherapies, in particular the bispecific antibody. The target for a lot of these immunotherapies is something called BCMA, or B cell maturation antigen. BCMA is a member of the TNF receptor superfamily, which is basically a receptor that is expressed universally on plasma cells and myeloma cells. So because of that, the effects of these immunotherapeutic agents is very targeted to myeloma cells. And the role of BCMA on the myeloma cells, it's involved in proliferation and survival of these plasma cells. So when you target them and attack them, it leads to cell death, which is the goal of the treatment. So how have we approached this? So there's been several different mechanisms in which you can target BCMA. As I mentioned, we have two approved CAR T cell products, and this includes cilta-cel or Carvykti or ide-cel or Abecma. These are now widely used in patients that have had 4 or more lines of therapy. We also previously had an approved agent called belantamab mafodotin, or Blenrep. This is an agent that was also approved in this space. Its mechanism of action, although it targets BCMA, it is an antibody drug conjugate drug. So it's not necessarily the same mechanism as we see with the CAR T cell product, but this was something that was used and recently was taken off the market by the FDA, mainly due to a confirmatory trial that was not positive, but trials are still ongoing in this with this agent in combination. So we will wait to see some more results to see if this will emerge as another option for patients. And then on the right hand of this slide, you see the bispecific antibodies. And again, this is another mechanism using T cell therapy to treat and target BCMA and treat patients with multiple myeloma. So here is a list of, again, various approaches. So on the left, you have the antibody drug conjugates, as I mentioned. Belantamab mafodotin was the first approval in that class. CAR T cell therapy, as I mentioned, we have two approved products. And then on the right, we have now one approved product with teclistamab, but there's several more under development with various different companies, again, using that mechanism. Majority of those are targeting BCMA. So how do these therapies work? So bispecific antibodies, as it sounds, have essentially two targets. So they find BCMA on multiple myeloma cells and kind of latch onto that. And on the other end, they target T cells. So CD3 is a marker on T cells. So what happens is, when you receive this drug, it'll go find BCMA and it'll go find CD3 and essentially bring the T cells over to the myeloma cells. And what that does, T cells are part of our immune system, so they're responsible for killing a lot of these tumor cells. So now you've essentially brought the T cell over to the myeloma cell. CAR T cell therapy also has a similar mechanism that it's using the T cell therapy to attack myeloma cells. But the difference between these two agents is that CAR T cell therapy requires that we first collect T cells out of your body. So, it's a T cell extraction. And then these T cells are genetically modified to express a receptor on the ends of them that is then going to target BCMA. So the process for having CAR T cell therapy is a lot more involved than bispecific antibodies because you've had to have the cell collection Then you have the manufacturing part, which usually takes somewhere between 4 to 6 weeks for those cells to be made. And then it requires 3 days of chemotherapy to help these cells grow. So kind of suppresses the immune system to allow these cells to grow when they're inserted. And then these cells are inserted, and then you have the mechanism that's described here. So in a way, CAR T cell therapy is sort of one and done. So you go through this whole process and you get the infusion. However, bispecific antibodies are a lot easier because they're off the shelf, meaning that you can just have them. You don't have to have the product be made, etc. But it is more continuous therapy because you require this medication to be given continuously to utilize this mechanism that's on this slide. So teclistamab, as I mentioned, is the first approved bispecific antibody in multiple myeloma. This, as I mentioned, targets BCMA and CD3. The trial that led to the approval is something that we're going to cover now. And this is, as I mentioned, is now FDA approved for patients who have had 4 or more lines of therapy. So the MajesTEC-1 trial was the one that led to the approval of this agent. And this was a phase 1/2 study. And this was looking at various doses of teclistamab and schedules of teclistamab and in patients that have had more than 3 prior lines. So including a proteasome inhibitor, immunomodulatory drug, and a CD38 monoclonal antibody. The dosing of teclistamab is basically, it starts with what's called step-up dosing. So you give very small doses, couple of days apart to see to mitigate really the effects of cytokine release syndrome, which is one of the side effects of T cell therapies. So it requires hospitalization, so approximately 1 week hospitalization to receive some of these step-up doses. And then after that is done and you've kind of gone through the process of the window of CRS or cytokine release syndrome, then after that, this is given weekly. So here were the patients that were enrolled. It was 165 patients in total. And as expected from trials like this, these are patients that have exhausted all of our other available therapies. So essentially, all the patients have had those prior classes of drugs, as I mentioned, the immunomodulatory drugs, proteasome inhibitors, and the CD38 monoclonal antibodies. And about 3/4 of patients in this study had more than 4 lines of therapy. So this kind of fits with what the FDA label is for this trial. And when you look at all comers, the overall response rate, so in how many patients did this, led to, killing of the myeloma cells, it was about 63%. So, about 2/3 of patients responded to this therapy. And if you look, if you dig a bit deeper, about 40% of those patients had what we call a complete response, so complete disappearance of the M protein in the serum and the disease from the bone marrow. And in terms of how long that response lasted, the median duration of that response was about 1.5 years. So it was about 18.4 months in patients that responded. And this compares favorably to what we see with CAR T cell therapy. So when you kind of look across the different therapies, even though there's no head-to-head trials, you're starting to now piece together some of the data we know from CAR T cells. And now this looks quite similar in some ways in terms of how long these therapies last. And then progression-free survival, which is, sort of the main thing we look at. What this basically means is at what point did patients, essentially have growth of their disease requiring, a different therapy. It was 11.3 months in this study. And if you look at the original CAR T trials, particularly with ide-cel or the Abecma product, it was very similar. So approximately a year is what we have seen with the original CAR T product. So, you're again achieving this with the medication that you can receive instead of having to go through the CAR T cell process. Some of the other CAR T products, such as cilta-cel or Carvykti, seems to have a longer progression-free survival. But again, we don't have head-to-head trials. So on paper, it looks like that one may be a bit more efficacious. It has a higher response rate and it lasts a bit longer. But again, it comes with some of these caveats of toxicity and the processes. So, but overall, this falls in line with what we've seen so far with CAR T cell products. When you look at safety, obviously we look at, what it did in terms of the blood counts. You see some reduction in your white blood cell count as typical for these types of therapies. Remember, this does not require any chemotherapy to be given right before you receive this. About 72% of patients had CRS. So this is something that we do expect, which is why patients are hospitalized for monitoring. And then the thing we've been paying the most attention to, I would say, with this particular agent and to some extent class of agents is infection. So about 3/4 of patients have had infections, but about almost half the patients had what we call grade 3 or 4 infections. These are more serious infection, often leading to hospitalization. And some of these infections included COVID-related deaths. So this is obviously these trials were done in the pandemic. And then, we also have seen some more sort of rare infections. So, when we're meeting patients now and they're going on to these types of therapies, we're very mindful of this and are really, looking to see any ways we can mitigate the risk of infection. So we give a lot of times these immune globulin infusions to sort of beef up the immune system and other mechanisms and methods that we can do. And in particular, counseling patients that if they do have signs of infection, they need to notify us right away. Cytokine release syndrome, again, which is seen with both CAR T cell therapies and bispecific antibodies, the rates of CRS appear to be lower with bispecific antibodies, and notably the severe CRS, in which you have severe deterioration of your blood pressure or oxygen levels requiring, let's say, intensive care unit stay. Rarely do we see that with these bispecific antibodies. And now we have a lot of experience in mitigating or managing CRS. So we give these agents such as tocilizumab, which is a medication that we give to block CRS. A lot of times we're giving this a lot earlier now than we used to. So we're really preventing some of those more serious CRS episodes. So this is now approved. It's been, again, given to patients with four or more lines of therapy. Even though it's widely available, we've had, some issues because it does require hospitalization. So we have a long list of patients that are interested in these T cell therapies, both CAR T cells and bispecifics. But because we have to admit patients, we now have to plan well in advance. And again, they have to have had 4 or more lines of therapy. And that last line of therapy, is to stop working for you to be eligible for this. But this is something that, we're now very rapidly treating lots and lots of patients. And then there's development already looking at this with various different combinations, because this is an injection, essentially. You can combine it with some of our other agents, et cetera. So combinations may be even better than giving this by itself. But more to come on that. And then also giving it earlier. Right now, this sits in 5th line. There's trials already ongoing looking at it in patients with earlier lines of therapy. So this is something you'll hear a lot more about going forward, but very exciting to have this option available for patients in the clinic. I wanted to just highlight a few other bispecific antibodies in development. This was another bispecific antibody targeting BCMA called elranatamab. And again, this is with a different company, but essentially looking at the same mechanism and a similar agent in terms of targeting BCMA and using that bispecific antibody method. The dosing of this particular bispecific, similarly, a lot of these bispecifics will have these step-up doses where you give a small dose a few days apart to start off with to really kind of lower the incidence of severe CRS, and then go ahead with weekly dosing. And then after cycle 6, which is essentially 6 months, you end up basically receiving this every 2 weeks. So the schedule does taper off. And again, similarly, I didn't go into too many details of this study, but just so you know, it's kind of falling in line with what we've seen with teclistamab. So an overall response rate is about 61%, similar to what we saw there. So seems comparable. We don't have the progression-free survival data quite yet, but it looks like it's going to fall again into that similar range. So at 12 months, 58% of patients are progression-free. So, we'll see what the median is, but overall, this also looks quite favorable to what we've seen with teclistamab. Infections, again, as I mentioned, is something we're paying attention to. It was reported in 2/3 of patients here. The grade 3 or 4 incidence of infection was slightly lower, but again, you're seeing a fair bit of infections with this particular product as well. And again, some of these are more rare infections, such as these fungal pneumonias and some of these viral infections that we've seen. So going on to yet another bispecific antibody that I think will be, something that hopefully we'll have available in the clinic in the not so distant future. So this is called talquetamab. This time, though, it's not targeting BCMA. It's targeting something else called GPRC5D. And this has been a recent discovery by one of our colleagues, Dr. Eric Smith, who's here at Dana-Farber, where you've essentially identified a new target on the multiple myeloma cell. And this is, again, very universally expressed in plasma cells without much expression in normal tissues. So this makes it an ideal target for some of these therapeutics. And this is an agent, again, a bispecific antibody that targets this GPRC5D, and then the other end attaches to CD3. So now it's bringing T cells over to a different target on the myeloma cell. So they've been, doing this phase 1/2 study and looking at two separate doses. One is a weekly dose, one is an every 2-week dose. And this was an update at our most recent meeting, looking at some data between these two cohorts. And as you can see, the response rates of this are in the 70s. So again, looks perhaps a little bit better than even what we've seen with the BCMA bispecifics, but generally in that similar range. So on the weekly dosing, the response rate was 74%. On the every 2-week dosing, it was essentially similar. And again, these are all the patients that have a very similar profile in terms of having the lines of therapy that I've mentioned before, including some of our commonly used medications. So this was, very active agent in this space. And again, looking to see how long these therapies last, as we've discussed before. So on the 0.8 milligram per kilogram dose every 2 weeks, it's approximately a year. So again, looking very similar to what we see, with some of the other bispecifics. And if you achieve like these deep responses, typically it lasts even longer, right? So as we said, it was about 18 months with what we've seen with some of our other bispecifics, and we'll see what it comes of this. But it's exciting to have a different target because BCMA is targeted via many different agents, and it'll be nice to have an option, perhaps for patients that have had BCMA, and then they can go on to something like this in the future. In terms of toxicity, again, kind of focusing in on CRS, it's seen as it is with most of these agents. So you see it in about, 3/4 of patients, but again, severe CRS is very low. This does have some more unique toxicity. So there is some expression of this on skin and hair follicles and nails. So this does lead to sometimes rash and other skin and nail bed changes. So something to note for this particular target. And then patients have also experienced what's called dysgeusia, and that means taste changes. So, again, it's very unique to this target, and that's something we have to monitor now as these trials, mature, to see, how bothersome that is to patients, and can we do anything to lower those side effects as much as possible as these agents become available, because this has obvious quality of life implications. You want to make sure that even though it's working, patients are having good quality of life and are able to live while on these types of therapies. So more to come on that particular topic. There were some patients enrolled in this particular trial that had prior T cell therapies. So these were patients that, about 70% of them had prior CAR T cell therapy, and even some patients had prior bispecific antibodies. None of them were targeting this particular agent. So they were targeting, usually BCMA, which is what we have available. But despite that, we had about 2/3 of patients respond to therapy. So this is what gets us very excited because, as we have all these options available, we want to figure out, well, can you give this and then this right after that? Because they're all using T cells. So if you're using T cell therapy one after another, is that going to work even though you're using a different target? These trials, again, the numbers are small, but they're starting to show that it does work. Perhaps the numbers are a little bit lower in terms of the success rate, but I think it's exciting to know this. And I think this is an area that we're going to have a lot more data over the next couple of years to know exactly, how to sequence some of these therapies. And then finally, the last bispecific that's in development, led by my colleague here, Dr. Jacob Laubach, and some of the clinical trials, is looking at cevostamab. So cevostamab is now targeting yet another target on the myeloma cells called FcRH5. And again, this is a bispecific antibody that latches onto that on the myeloma cell. The other end goes to the T cells and brings those two together. And these results have been updated over the last few years. And again, we're seeing, these are in dose escalation, dose finding type studies. And you're seeing, again, about approximately 2/3 of patients respond to therapy, even using this particular target. And, again, duration of response seems to be in that 1-year mark. So, it's nice to have all these other simultaneous development of these agents with bispecific mechanism to be able to use different targets on the myeloma cells. So a lot going on in this immunotherapy space, which is very exciting. So here's just a summary slide. Majority of the data is being generated with BCMA bispecifics, and now we have one available in the clinic, which is fantastic. We may have more BCMA bispecifics available, and then we'll have to see the pros and cons of either approach and see if they're different enough for us to choose one over the other. So more to come on that. And they're also looking at different schedules to make it easier for patients to receive that, when they're on it. So perhaps they don't need to get it every single week. Maybe we can, lower it to every 2 weeks or even monthly, et cetera. And then you have these other targets on the bottom: talquetamab targeting GPRC5D and cevostamab targeting FCRH5. So open questions in 2022 and beyond, now in 2023. So what's the optimal method to deliver these BCMA therapies? Should it be CAR T cells? Should it be bispecifics? CAR T cells tend to have higher response rates, but because of availability, manufacturing, toxicities, it's not something that we've been able to give to every single patient that is interested and eligible because of supply chain issues and just the logistics of it all. And a lot of times you end up having patients that need more immediate therapy and they can't wait for these cells to be made and go through this process. So it's got its own challenges, but does seem to have perhaps a bit higher activity particularly with the cilta-cel or CARVYKTI product. Belantamab mafodotin, again, this was the ADC or antibody drug conjugate that was previously available, but it did have the side effect of keratopathy, which is, some vision changes that patients were noticing. So, we had become pretty good with managing this with the eye doctors, et cetera. But, and then it's no longer on the market. So we hope it'll be back with, a different, as some of these other trials that are ongoing, looking at different combinations, et cetera. So more to come on that. That's also a nice option because it is readily available. So hopefully, we'll be able to offer that to patients in the future. And then again, these bispecific antibodies, I think they may strike the ideal balance. They're off the shelf, they're outpatient. Eventually, after you go through the initial phase, tend to have lower toxicity than what we've seen with CAR T cells. And you can use many different targets, as we just discussed, instead of just using BCMA. And then we've talked about, can you administer one BCMA therapy after another? Right now, we don't have great data. We have some of these small trials and cohorts to tell us that, yes, it can still work, but, we have to still see what the best approach there would be. Like, for example, if somebody got CAR T cells and now their disease is progressing, a year or two later, can you give them teclistamab? It's something we're doing now in the real world as we're treating some of these patients, but we need to generate a bit more data to know the exact effectiveness of that approach, or vice versa. If you get bispecific antibody, can you then get CAR T cells? So I think that's an area that we have to, generate a bit more data. And then my hope is, we can give these BCMA therapies early. Right now it sits in fifth line, which is, a lot of these patients, as you probably are aware, are becoming refractory to some of our approved agents way earlier than fifth line, because we're giving 4-drug, induction therapies. We're giving maintenance therapy now that includes several medications. Then they usually go through some of these other more approved combinations in second and third line. So a lot, you're looking for these types of therapies way earlier. And on top of that, these are immunotherapies. So the immune system is a lot healthier earlier in the disease course. And perhaps we can, utilize that immune system even better if we give it in earlier relapses or even in patients with newly diagnosed disease. And there are some head-to-head trials ongoing looking at CAR T cells versus stem cell transplants. So there's, it's being studied across the range of myeloma disease. And over time, I think we'll have a little bit more clarity as to kind of where this will be fitting in most optimally. So in conclusion, these is really exciting to have these immunotherapies available. They're both great options in terms of CAR T cells and bispecifics. As we talked about, limitations do exist with each approach, but teclistamab so far has been really the most convenient because of its just availability and the outpatient dosing that we can do after the initial step-up dosing. We still need to understand better the mechanism of resistance to these T cell therapies. And then we have new targets under study, as I mentioned, GPRC5D and FCRH5, combination therapy, and trials in earlier lines are ongoing, which will then give us a better sense of optimal sequencing. And with that, I want to thank you for your attention. Take care.